Hello everyone, welcome to my YouTube channel. Today I am going to discuss about Krebs cycle. This is a kind of metabolic pathway, which is a catabolic kind of pathway that ATPs be made from food molecules such as carbohydrates, proteins and lipids. This is a cyclic pathway, that is why it is called Krebs cycle. It is a very essential pathway that food molecules such as proteins, lipids, and carbohydrates can be converted into ATP when you compare with glycolysis, which is the pathway for only carbohydrates. This one is very essential. And again, when you compare with the glycolysis, which is a catabolic pathway, this pathway is, the Krebs cycle is very essential that per one glucose molecule, there will be extraction of a lot of ATPs. If you are new to my YouTube channel, please subscribe. I am very grateful for that because it is a big energy, a lot of energy like Krebs cycle. Thanks so much. It is also called TCA cycle or citric acid cycle. Why it is called TCA cycle? The TCA cycle, TCA is the abbreviation for tricarboxylic acid cycle. Tri means 3. So there are 3 carboxylic groups in the first molecule which made in the process. That is citric acid or citrate. Therefore, it is named as citric acid because the first product in the cycle or in the pathway is the citrate or citric acid. It is named Krebs cycle because the discoverer of the pathway was a scientist, Hans Krebs. From foods we eat through metabolism, there will be extraction of ATP. The foods are just lipids, carbohydrates, and proteins. When we consider carbohydrates as glucose as an example, there will be conversion of one glucose molecule into 2 pyruvate molecule through the process called glycolysis. This is a kind of pathway. You can look at my video on glycolysis. This is one of the catabolic pathway. Then these 2 pyruvate, after it made from glucose, where it will go. So the fate of the pyruvate is, one, it will be converted into acetyl-CoA. Acetyl-CoA and enter into TCA cycle. This is very essential step because when it converted into this acetyl-CoA and enter into TCA cycle, it will make a lot of ATP. In this process, in the conversion of 2 pyruvate into acetyl-CoA, there will be a removal of 1 carbon through carbon dioxide molecule. The process is called decarboxylation. And again, here there is one coming in of CoA molecule. Therefore, you can see here CoA. And the enzyme which involves in this process is pyruvate dehydrogenase. Or there are a lot of enzymes, we can call them pyruvate dehydrogenase complex. Again, here there is a synthesis of NADH molecule. This is a citric acid cycle. Here, acetyl-CoA is here, and then oxaloacetate will be there, and acetyl-CoA plus oxaloacetate, this is the first step, and it will make citrate. Acetyl-CoA is 2 carbon compound, oxaloacetate is 4 carbon compound, then 2 carbon plus 4 carbon, it will be 6 carbon. Therefore, citrate is a 6-carbon compound we can call citric acid. Therefore, the first product here is the citrate or citric acid. Then citrate will be converted into isocitrate. Isocitrate will be converted into alpha-ketoglutarate. In this step, there is the conversion of the NAD molecule, which is oxidized. This plus indicates this molecule is oxidized. Then it will be reduced into NADH molecule. Again, here is removal of carbon through 1 carbon dioxide molecule. Therefore, from 6 carbon, there will be 5 carbon. The next compound will be 5 carbon compound. That compound is alpha-ketoglutarate. Again, in this step, alpha-ketoglutarate will be converted into succinyl-CoA, and here also there is a reduction of NAD molecule. This one is oxidized, then it will be reduced into NADH molecule. Again, here will be coming in of acetyl-CoA. This is acetyl-CoA. Then here is decarboxylation again, the second decarboxylation, or the second removal of carbon is through carbon dioxide molecule. Then from 5 carbon, the next compound will be how many carbon? 4 carbon compound. Therefore, succinyl-CoA is 4 carbon compound. Again, succinyl-CoA will be converted into succinate. In this process, there will be making of GTP from inorganic phosphate. GDP plus inorganic phosphate. This is Pi. I indicates inorganic. P indicates phosphate. Therefore, inorganic phosphate plus GDP give us GTP. Here will be removal of the acetyl-CoA at this step. Then succinate will be converted into fumarate. The compound after this succinyl-CoA will be 4 carbon compound. Here you can see 4C, 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 4C. In this step, in the conversion of succinate into fumarate, there is making of FADH2. Okay? In this process, making of FADH2 from FAD molecule. This is flavin adenine dinucleotide, and the NADH is, or NAD is, nicotine amide adenine dinucleotide. Here, fumarate will be converted into malate in the 7th step. Here is coming in of water. Water is important. And malate will be converted into oxaloacetate. Again, there will be remaking of oxaloacetate, or the cycle will continue again. Okay, therefore, oxaloacetate will be always there for the process. In this step, in the conversion of malate into oxaloacetate, which is the last step, there will be reduction of NAD molecule. Here, this oxidized NAD is reducing into NADH. When we say reduction, if a molecule gets hydrogen, we say that molecule is reducing. Therefore, this is a cyclic process. There are 8 metabolites and 8 step process. Here we can see enzymes which involves in each step. We will see in the next few minutes. Here, these are enzymes in each of the steps. For example, when oxaloacetate and acetyl-CoA convert into citrate, the enzyme is called citrate synthase. It says synthase because citrate synthase. Because what molecule is here? It is citrate. So synthase means from the name synthesizing. So citrate molecule is synthesizing. So the name says synthase. Then citrate will be converted into isocitrate. The enzyme will be aconitase. When isocitrate ketoglutarate, the enzyme is isocitrate dehydrogenase. Here is dehydrogenase. In the 3rd step, in the 4th step, in the sixth step, and in the last, that is eighth step, there is enzyme called dehydrogenase. That indicates these steps are oxidative oxidation reduction process takes place. There is oxidation reduction in this step. Therefore, isocitrate dehydrogenase, alpha ketoglutarate convert into succinyl-CoA. The enzyme is alpha ketoglutarate dehydrogenase. Succinyl-CoA converts into succinate. Then the enzyme will be succinyl-CoA synthase. When succinate converts into fumarate, the enzyme is succinate dehydrogenase. Fumarate convert into malate, the enzyme is fumarase, and malate convert into oxaloacetate, the enzyme is malate dehydrogenase. Therefore, you can go back and look, there is redox reaction in these 4 steps. For example, in this third step, there is redox reaction. Here is reduction of this oxidized NAD molecule in the third step. In the fourth step, the same. In the sixth step, again, here is FAD oxidized form is reducing into FADH2 molecule. In the last step, there is reduction of the NAD molecule into NADH molecule. Therefore, this is where the dehydrogenase enzymes are involving. Dehydrogenase means removal of hydrogen. Net result of the citric acid cycle: acetyl-CoA plus 3 NAD oxidized form. This plus indicates this nicotinamide adenine dinucleotide is in the oxidized state, plus FAD, which is again oxidized form, plus GDP plus inorganic phosphate. This I indicate inorganic. This indicates phosphate. So inorganic phosphate plus 2 water molecule, then to the product side 2 carbon dioxide plus 3 NADH plus FADH2 plus GTP plus CoA plus 3 hydrogen ion. When you go back and, for example, when you go back in the cycle, here you will see those molecules. In the first process, there is water is coming in. This arrow indicates this molecule is coming into the process. It is entering. And this kind, the outgoing arrow indicates this molecule is leaving the pathway. Therefore, here the CoA, this molecule is CoA attached with acetyl. Its role is bringing this acetyl part to the process. After carrying and bringing, it will leave the pathway in this way. Considering the net pathway, here is 1 water molecule entering into the process in the first step. Again, here is as the 7th step. Here there are 3 NAD oxidized molecules at this step, 3rd step, 4th step, and as the 8th step, 3 NAD molecules. And reduced NADs are also 3. Again, here is acetyl-CoA. 1 acetyl-CoA is in the formula because this acetyl-CoA is not in the pathway, because after carrying and bringing, it will leave the process. Only this will be considered in the chemical formula of the citric acid cycle. Here is 1 GTP formation from GDP and inorganic phosphate at 5th step, and here is 1 FADH molecule which is oxidized, converting into reduced FADH molecule. So these are the molecules which you can consider in the net pathway, okay, as a net result of the citric acid pathway. Where do all the NADHs and FADHs go? The answer is into electron transport chain, abbreviated as ETC. This is collection of proteins. electron transport chain is a chain or collection of proteins. We will see this chain of proteins in the next few minutes. Here is starting of catabolic process from glucose. Then glucose enters into a cell. This is a cell. Then through the glycolysis, there will be conversion of glucose into pyruvate. Then what will be the fate of pyruvate? There are two fates depending on the presence of oxygen or absence of oxygen. If there are oxygen molecules, then this pyruvate will enter into the mitochondria. Then the pyruvate will be converted into acetyl-CoA, and acetyl-CoA will enter into TCA cycle. Then there will be production of NADH, FADH2, and these FADH and NADH2 are the energy carrier molecules. That means there are a lot of ATPs within these molecules. The energy they are carrying is in the form of electrons. That is why electron transport chain is important. The other fate of this pyruvate is fermentation, through which the pyruvate will be converted into lactate. One thing we have to consider here is there should be oxidized form of NAD always there, because there will be energy synthesis in cells, because living organisms always need energy. Therefore, here is a NADH formation in the glycolysis. If there is oxygen, then there will be oxidation of this NAD in the mitochondria, and there will be free NAD molecule. If there is no oxygen, again there should be also free NAD molecule. That is important. Then there is a mechanism of freeing this molecule, and then it will go back and used as NAD oxidized form in the glycolysis. This one is a mitochondria. There are two membranes, outer membrane and inner membrane. The space between these two membranes is called intermembrane space. The TCA cycle takes place in the innermost of the mitochondria we call matrix. In the TCA cycle, we know that there is formation of NADH and FADH2. Then there will be releasing of energy in the form of electron from these molecules, and then these electrons will enter into this inner membrane of the mitochondria. This is where the electron transport chain or the protein complex found in the inner membrane of the mitochondria. Then electron will be transported through this membrane. Then this is transporting. Finally, the electron acceptor will be oxygen. Then there will be formation of water. Then when electrons are transporting through this membrane, then there will be pumping of protons, that is hydrogen ions, into the intermembrane space. Then a lot of hydrogen ions or protons will be in this space. Then there will be electron gradient between the intermembrane space and the matrix. Then there will be what? Coming back of these protons into the matrix. This is through which the ATP will be made. The movement of these protons is through the proteins, which is also an enzyme called ATP synthase. Therefore, there will be making of ATP in this process that we call chemiosmosis. Here is NADH will enter into the mitochondria. The NADH is from glycolysis and the conversion of pyruvate into acetyl-CoA will enter into the electron transport chain and then making of ATP in the process. Some important notes: one NADH will make 2.5 ATPs and one FADH2 will make 1.5 ATPs. This is because one NADH, when one NADH is there, it can pump 10 hydrogen ion here. Okay, 10 hydrogen ion can be pumped into intermembrane space by one NADH. Then 4 hydrogen ion can make one ATP molecule. So from 10 hydrogen ions, that is from one NADH molecule of 10 hydrogen ions, there will be 2.5 ATP. And one FADH molecule can pump 6 hydrogen ion. Therefore, 6 divided by 4, because 1 ATP will be 4 hydrogen ion, it will be 1.5. That is the concept behind 1 NADH is equal to 2.5 ATPs and 1 FADH2 is equal to 1.5 ATPs. So electron transport chain is the final step in the breakdown of glucose. The first step is glycolysis, then the next step will be the TCA cycle, and the final step will be the electron transport chain. It is also the point at which most of the ATP is produced. High energy, we will see this point. High energy electrons and hydrogen ions from NADH and FADH2 produced in the Krebs cycle are used to convert ADP to ATP. So this is just the inner membrane of the mitochondria. These are the protein complexes or the electron transport chain, through which electrons from the NADH and the FADH molecules will be transported, and then finally there will be electron acceptor molecules, that is oxygen and formation of water, and then this is ATP synthase, a protein on which ATP will be made. So there will be 32 ATP making from one glucose molecule through the electron transport chain. As shown in above figure, NADH and FADH2, the energy carriers are converted to NAD and FAD. Electrons move along the mitochondrial membrane from one protein to another, that is in the electron transport chain. Hydrogen ions move from mitochondrial matrix to inner membrane space, and hydrogen ions then diffuse down their concentration gradient back from the intermembrane space to matrix, across the membrane and into the matrix through ATP synthase molecules in chemiosmosis. This is where the ATP making takes place. Oxygen is the final electron acceptor in the electron transport system in cellular respiration. Protons and electrons are transferred to oxygen to form water. So electron transport chain, here is a cytosol. This part is a cytosol inside the cell, and this one is a mitochondria, and here the glycolysis takes place in the cytosol, and then glucose converted into 2-pyruvate, then it will be converted into 2-acetyl-CoA from one glucose molecule. There will be 2-pyruvate molecule and then 2-acetyl-CoA molecule. The acetyl-CoA molecule will enter into Krebs cycle, and from the glycolysis there will be NADH formation. We know that this NADH will enter into the electron transport chain. The electron released from the NADH will enter into the chain. Again, in the conversion of pyruvate into acetyl-CoA, there is also NADH, so it will also enter into the electron transport chain. The FADHs and NADHs from the TCA cycle also will enter into the electron transport chain. 6 NADH and 2 FADH2 pair, 1 glucose molecule. Again, 2 NADH from 1 glucose molecule in this process, conversion of 2-pyruvate into acetyl-CoA. Again, 2 NADH from the glycolysis. Just look to the pathway that these molecules are there. This is just for 1 glucose molecule. Then all these will enter into electron transport chain and converted into ATP. So this one is just, it says 38 ATP. That this is because considering 3 ATP from one NADH molecule and 2 ATPs from one FADH2 molecule. Just the recent discoveries show that there are 2.5 ATPs per one NADH molecule and 1.5 ATPs from one FADH2 molecule. Actually, some scientists like to round up the 2.5 to 3 and 1.5 to 2. So don't worry for the number, and finally we will see the calculation and we will get the 32 ATPs from the one glucose molecule. So how many ATPs per glucose molecule? That means from one glucose molecule, how many ATPs can be made? There are two ways of phosphorylation. Phosphorylation means adding phosphate, and the two phosphorylation processes are substrate level phosphorylation. When we say substrate level, in the pathway there are substrates, the metabolites. Therefore, from the conversion of one metabolite to other or one substrate to other, there are some ATP making processes. That is substrate level phosphorylation. The other is oxidative phosphorylation. This is just the phosphorylation or adding of phosphate to ADP to make ATP after electron transport chain or in the electron transport chain. In the substrate level phosphorylation, for example, from glycolysis, there are 2 ATP net. We know that there are 4 ATP making in the process, but 2 are consumed in the first steps. Then you should subtract that consumed one. Then you will get 2 ATP. That is through substrate phosphorylation from glycolysis. Again, in the conversion of pyruvate into acetyl-CoA, there will be 2 NADH. This is just for oxidative processes. Again, here 2 NADH from glycolysis. We know that they will be converted into ATP after or in the electron transport chain. Then from the Krebs cycle, there will be 2 GTP from substrate level phosphorylation, which is equivalent. This is equivalent symbol or equal symbol. 2 GTP is equivalent to 2 ATP. Energetically, they are equal. Therefore, when we add 2 ATP from glycolysis and 2 ATP from Krebs cycle, you will get 4 ATP from one glucose molecule through substrate level phosphorylation. Then the oxidative phosphorylation will take place in the electron transport chain, and let's look at it. ETC means electron transport chain. So we know that one NADH will make 2.5 ATPs, one FADH2, 1.5 ATP. We know the reason. We have discussed that because 4 hydrogen ion will make 1 ATP and 1 NADH will pump 10 hydrogen ions. So 10 NADH molecule will be made from one glucose molecule when you consider the NADH from glycolysis, the NADH from conversion of pyruvate into acetyl-CoA, and the NADH from Krebs cycle. You will get 10 NADH molecules. Then 10 times 2.5, you will get 25 ATP. 2 FADH2 from Krebs cycle from one glucose molecule. From one cycle, 1 FADH2. From one glucose molecule, there are 2 pyruvates. Then there will be 2 acetyl-CoA. Therefore, there will be 2 cycles, 2 Krebs cycles for one glucose molecule. Therefore, we are considering that. In this case, also the same. And therefore, 2 times 1.5, which is 3 ATP, 25 plus 3, 28 ATPs. Is through what? Oxidative phosphorylation. Therefore, 4 ATPs from substrate level phosphorylation, 28 ATPs from oxidative phosphorylation. There will be 32 ATPs from one glucose molecule. That is all. That is all. Thank you so much. Please don't forget to subscribe to my YouTube channel. Have a nice time. If you like this video, please genuinely like my video. Bye bye. Have a